Thank you very much. It's a great pleasure to be here and very nice to take part in the second AI3SD event within a relatively short amount of time. So it's nice that apparently these are topics where we're quite well aligned. What I would like to talk about during the next plus minus 30 minutes is something which I believe is close to all of our hearts, which is that we desperately need new and better therapies. We all agree that cancer, heart disease, neurodegeneration, and many others desperately require better therapies, better than the ones we currently have available. And one way of doing that, and probably one of the most essential way of doing that, is drug discovery. So on the next slide, we're looking at a very brief overview of what drug discovery actually looks like. So when we see the drug discovery funnel, it starts on the left-hand side with the target idea, the target hypothesis. And then we basically move through the various stages, starting with target to hit. We're saying we have an idea for a target that could be, for example, a gene and its gene product. And now we want to find a hit, some very first chemical matter that is the starting point for what later on can become a drug. And then we move from a hit or from various hits into leads. We optimize those leads. And at some point, we then basically reach the preclinical stage, where it's often about ascertaining whether we can see promising data in other model organisms, for example, in mice. That is also the point when there is the famous IND filing, and the IND filing then is the entry ticket into clinical trials. And as we know, basic clinical trials within phase 1, 2, and 3 then make sure that anything which we use in humans and which we want to bring to market actually is safe, is effective, and is better compared to what we currently use to treat a certain disease or condition. I think it's really important to always think about this funnel when we talk about drug discovery and trends in drug discovery, because this is something which is very, very basically important and very important for the understanding for what the process looks like and for how the process in parts is really very slow and in parts can be improved upon by using technology. But before that, let's basically look at some numbers regarding those various stages. I don't want to go into too much detail, but it is very important to note that each of these stages is really expensive. It takes a lot of money to take compounds and take chemical matter through these various stages. And as we can see from the very first line, the likelihood of something making it to the next stage is definitely less than 100%. So we talk about a process where at every stage we're losing quite a substantial amount of potential drugs. And this then means that at the very end, we only arrive with one compound for maybe 10,000 that could have been tested earlier. What it then means is that this whole process just for one single new drug can easily cost more than $2 billion. So we talk about a huge amount of time, cost, money, resources that goes into a single new drug being approved. I think it's also very worth pointing out that we're moving more and more into an environment where precision medicine or personalized medicine or individualized medicine, all of these are often used interchangeably, is the paradigm that we want to follow, where we want to define patient populations far more narrowly, far more stringently, and we want to develop new drugs that really fit a particular patient population. And that is something which will require a very different approach to drug discovery, because that would mean we want to develop 10 times, maybe 50 times the number of drugs we currently have. And we therefore need to rethink the way we approach drug discovery. Another reason why we have to do that is because the model I just showed is something which over the years did not become better, but it became worse. There is something called Irum's law, in reverse of Moore's law, that describes how productivity in terms of number of drugs being approved per research dollar has been declining over the past decades. We can also imagine that as we're moving lower and lower on this trajectory, at some point we're reaching very dangerous territory where developing a new drug is no longer financially financially sustainable and is something which for biotech companies, pharma companies can become too risky to undertake. So in other words, we need to stop this trend, we need to reverse this trend, and we need to find a way where drug discovery can become more productive again. However, as with everything, you want to understand first what is wrong before you can then kind of apply a remedy. And so in this case, if we look at what potential underlying causes for this loss in productivity and for this decline in productivity are, there's primarily two things coming to mind: speed of data generation and quality of data generated. In terms of quality, there have been studies conducted, for example, with Bayer and Amgen over the past few years that showed very clearly that only a small fraction of peer-reviewed published research findings are actually reproducible. The numbers are scary. The data kind of that we have shows that only 10 to 20% of all published peer-reviewed findings, in this case primarily within oncology, could actually be reproduced, even when contacting the original authors, even when trying very hard to make these things work. So in other words, we're talking about an environment where data quality, data annotation, data structure is not the way it's supposed to be. And at the same time, we still conduct research in very much the same way we've been doing research for the past decades. If you look at labs today and labs 20, 30, 40 years ago, they look practically the same. Of course, now we have PCR machines and we have better microscopes, but the work mode itself is still the same, where researchers spend 60, 70, 80% of their time manually pipetting small amounts of liquids and spending only a fraction of the time reading, thinking, hypothesizing, discussing results, etc. So in other words, the drug discovery ecosystem right now is in a stage where it is necessary to think about how we can rethink in a very basic way how research happens and how we go about discovering new drugs. What the next basic graph shows is a schematic of the drug discovery ecosystem, and where we can basically try to put different companies and different players into boxes or categories. In this case, in the bottom left-hand corner is the traditional players, the way kind of all of us know them. Biotech companies, pharma companies, quantitative research organizations. We could add universities there as well. With a low degree of automation or hardly any automation and a low degree of data mining or AI capabilities, sometimes even non-existent. This is the way we've been doing research so far, and this is the way it has been done, and in many cases continues to be done in many of these organizations. But at the same time, we can see that this ecosystem is evolving right now in front of our eyes very rapidly. One of the key techniques is AI. This is just a very small selection of a few headlines over the past maybe 2-3 years, where we've seen that AI-driven drug discovery companies have made the headlines by partnering with pharma companies, by generating new insights at a formidable speed, and by doing things that others in the industry for a long time considered impossible. A few of these players include in silico medicine, e-therapeutics, Exscientia, Benevolent AI. So those are the companies that really make a difference in this field by applying AI to drug discovery. At the same time, it's also really interesting to note that this trend towards AI is accelerating. This basically tracks the number of AI drug discovery companies and AI machine learning companies founded over the past few years, where we can see a very steep rise. And this is something which is incredibly interesting because AI, as we all know, kind of has evolved in various phases. And right now we're seeing a tremendous interest in applying AI and machine learning tools within drug discovery. And we're seeing a lot of companies and more companies doing that. So what we can then do is we can then map these companies in the schematic we've just seen, where the AI-driven companies, Atomwise and Silico, Benevolent, basically can be put kind of into the top left-hand quadrant, where data mining and AI capabilities play a substantial role for these companies. So what can AI be used for within drug discovery? The short answer is for almost everything. This list is taken from Simon Smith, who is the Chief Strategy Officer of BenchSci, an AI-driven company in the drug discovery space itself. And Simon Smith's list is probably one of the most important reference points if you want to learn about AI companies within drug discovery. And his list contains several hundreds. I mean, this list was updated several times. Includes several hundred companies that apply AI and machine learning technologies in different parts of the drug discovery ecosystem, starting with aggregating and synthesizing information via data generation, via, for example, de novo drug design, via drug repurposing, down to things such as optimization of clinical trials and clinical trial recruitment or real-world evidence. In other words, across the whole breadth of the drug discovery funnel that we've seen in the beginning, across the whole bandwidth of different processes, there are companies, there are researchers suggesting and using AI solutions for parts of the process. I just want to highlight one just to give an example, which in this case is de novo drug design, where there's been a few very interesting papers coming out of the past few years. And one of them is this one here, published by Alex Voronkov and his group pharmaceutical medicine, who spoke about enabling the rapid identification of, in this case, a DDR1 kinase inhibitor, where they completed a process that normally would take between 1.5 and 2 years in about 45 days. This was published in Nature Biotechnology last year, and this is, I think, a very interesting paper, and one that really shows how, just giving this one example, artificial intelligence can really have quite a tremendous impact on drug discovery processes that for a long time we assumed to take a certain amount of time and a certain amount of effort, and that we can now suddenly see, thanks to new technologies, we might be able to make something work that, again, for a long time we believe was impossible. So if we move back to the schematic, what we've just seen is that there's a lot of companies and some very exciting companies in this top left-hand quadrant. And of course, now you're kind of wondering what is happening with the other two. And so what we can see here is that there's quite a few companies using automation to different degrees, to different purposes. So in other words, the right-hand side quadrants are for the companies using automation in many different ways. There's, for example, companies within synthetic biology, such as Ginkgo Bioworks and Zymergen. There are the cloud laboratories, primarily Strateos and Emerald Cloud Lab in the US, and ourselves from Arcturus in the UK. And there's companies that don't use automation to power other users, but they use automation in-house for their own drug discovery efforts, such as Recursion Pharmaceuticals and Incitro, both in the US. Of course, everybody who works in the industry will know that robotics and automation is something which has been around. Pharma companies in particular, the ones kind of with deep enough pockets, have used automation in the past. However, what they used robotics and automation for was primarily high throughput screening, a process that enables you to, for example, find the first hits. The first hits then are the starting point to develop new chemical matter, develop a new drug. High throughput screening has been automated because we talk about screens involving 500,000, 1 million, sometimes even more molecules. Only machines can do that type of work. However, these processes are very rigid, linear processes where there's a single experiment done over and over and over again. And these facilities and these types of robots have never been designed to do more than this one type of experiment. So it's very difficult, if not impossible, for these robots to do anything different but this one type of screen. What now has been happening is that over the past few years, and this is less than 10 years, life sciences and laboratory automation has made huge steps towards being able to automate a far broader range of discovery work, a far broader range of functions, including hit-to-lead work, including lead optimization, partly even including preclinical work. There's some very interesting work on, for example, automating zebrafish. There is interesting work on automating worms. There is some interesting work on, for example, having vivarium settings where animal work can be conducted. in a far more traceable fashion. So in other words, across the whole pipeline of drug discovery, anything from idea towards IND is something which is now touched by automation, and automation plays more and more of a role. And we also can see this kind of reflected in different news articles. For example, In Sitro, led by Daphne Koller, she was the first female machine learning professor at Stanford's, founder of one or two very exciting companies in the cloud space, and then later on founder of In Sitro. There's news about Ginkgo Bioworks. There's Recursion Pharmaceuticals. There's collaborations between Stratios and Eli Lilly. And of course, there's also kind of our own collaboration within silico medicine that I've mentioned before. In other words, we can see that the same way that AI is creating headlines within drug discovery, the very same thing is also true for robotics. And I think the reason is quite clear, because once we look at what robotics can actually do, we can see that automating experimentation is something which has tremendous impacts on cost and speed of data generation and on quality of data generation. And those are probably the most important metrics you could potentially apply to look at what impact a solution has. And so if we look at those in more detail, we can see that, for example, the cost it takes to profile a drug or to move from one specified stage to another specified stage is something which, thanks to automation, can be drastically reduced. At the very same time, generating new data is something which often requires months of troubleshooting to get something up and running. Anyone who's been doing a Western blot knows how painful it can be to optimize a new antibody. At the very same time, once you use automation, once you're able to use a library of pre-established, pre-validated, pre-configured protocols, you can start generating data in a fraction of the time. When we then look at the quality that comes out of machine-generated data, we can also see that the data quality is something which plays on a completely different level. And the reason that is quite interesting, because as of now, within drug discovery research, we're only looking at the tip of the iceberg. So the tip of the iceberg is what everybody in a manual laboratory currently does and currently looks at. Data sets that are normally isolated, they're not connected. You might look at one experiment and you look at a different experiment, two different devices, maybe two different postdocs or two different researchers. There is no link, no connection between them. At the same time, methods and protocols are ambiguous. What do I mean by that? A scientific protocol only gives a certain degree of details. So a protocol might, for example, say mix the sample. But mixing the sample is not very specific. It could be shaking, it could be stirring, it could be vortexing, it could be many different things. Maybe for half a minute, maybe for 5 minutes, maybe at room temperature, maybe chilled. In other words, it's very difficult to generate robust data when working with protocols and methods that are so ill-defined. We also deal with storage systems that are often not state-of-the-art, where, for example, a USB stick might be home to really important data. And finally, data standardization and data format standardization is only the very beginning, which means in many cases it will be very hard to properly transfer data between different groups, or even within one group, simply because everybody is storing them differently. And now, what is the benefit that automation brings? Automation, especially when coupled with a wide range of sensors capturing relevant data, is something which means and enables data to be captured on a very different level. We can have a complete digital data capture. Instead of tracking things with pen and paper, where we write down incubation started at 8 p.m., done overnight, we would have a machine, a robot, an automated setup that collects a very broad range of different parameters 24/7. A setup that knows exactly when each experiment starts, when each experiment finishes. At the same time, these experimental protocols are fully standardized. For example, when it's about deciding whether the incubation is 8 hours or 10 hours, this is something which can be predefined, it can be specified, and will then be obeyed by the robotic system. Not having the ambiguity as to whether an overnight incubation, which could be anything between 7 and 12 hours. This also means there's full audit trails, which becomes very important when working in an industry that requires data to be of the highest levels of integrity and trust. It also means data is interoperable, which then becomes an interesting conversation when thinking about fair data principles: findable, accessible, interoperable, reusable, which should be the gold standard for any data generated in any field, but especially within biomedicine. And it also means that anything which goes into the system can be controlled far more tightly. One of the reasons why we have such a bad track record with reproducibility is the provenance of research reagents. We know, for example, that 1 in 2 cell lines being used in research is either contaminated or misidentified. 1 in 2 cell lines. Those are all things that we need to change, that we need to address before we can really think about bringing research into the 21st century. To make it a bit more visual as to what these differences actually mean in real life, I've brought kind of a very simple IC50 curve, standard biochemical assay, where we're looking at one inhibitor and comparing different doses of that inhibitor to find out what the IC50 is, what is basically the concentration where we can see 50% inhibition of a given target. In this case, this graph was generated using serial dilution. We only have 10 or 11 dose points, and there's a limited number of information we can gather from this particular experiment. At the same time, when looking at a fully automated approach, in this case from our own laboratory, instead of relying on a handful of data points and interpolation, we can generate 10 times, 50 times the number of data points. We can really track every single possible inhibitor concentration and actually generate a data point instead of interpolating between 1 or 2 of them. At the same time, we also get a range of additional metrics: experimental metadata, for example, timings, for example, work cell conditions, for example, details for that specific experimental run. And we're also getting kind of all the benefits of quality that comes from having lower standard deviations, greater precision, greater accuracy. So we can really see that there is a tremendous difference between a manual experiment and between something that was generated using a machine. What I would now like to do is I would now like to tie these two themes together, because we've seen also in the talk before how machine learning and artificial intelligence, to use the kind of the broader term, is something which is having a tremendous impact in the drug discovery ecosystem already. I've also highlighted how robotics and automation is a second extremely important trend that also has a huge impact on the industry. What happens if we tie those two together? Once we combine robotic data generation and AI, we arrive at a very, very exciting point, a very exciting inflection point. Because once we execute assays and experiments using robotics, we achieve full protocol adherence, full reproducibility. At the same time, we have data capture that's far more complete than anything we've ever done before. And we know that anything which comes in, anything which goes in, anything which comes out is trusted, validated, has been audited. In other words, we're arriving at a very rich and very valuable data sets. And those are, again, validated, they're comparable to one another, and they're mineable. And this is what makes it so interesting for machine learning applications, because we know that by now the biggest bottleneck is no longer machine learning algorithms, but the biggest bottleneck is getting access to high quality data sets. And this means with this combination of automation, robotics on the one hand and AI on the other hand, we can really see drug discovery entering a new era where training data is generated and model development and model validation then basically happens practically in real time. Because thanks to the advances in speed and acceleration that automation can grant us, we can really compress development cycles quite effectively. And what it then basically means perspectively is that we can think about this whole drug discovery process from idea and from target down towards IND to become far more scalable and something where the likelihood of finding new drugs is rising, because any decision made is based on high quality, trustworthy data. And any decision made can be augmented with AI because data is AI ready. To conclude, I would just like to show kind of a brief case study. In this case, something we've done with in silico medicine. This was the press release also which you showed briefly before, where during the beginning of the COVID-19 pandemic, in silico medicine used its machine learning approach to design new JAK inhibitors in this case, because they'd been shown previously to be of potential use against COVID-19 and patients already suffering from the disease. By the way, a study kind of done by Benevolent AI, which was a very interesting paper. So in silico medicine, basically took this idea and then generated new JAK inhibitors, synthesized them, and sent them to us, where we could provide full profiling, full validation, and generates a more than 1 million data point assay package within less than a week, which was then fed back into the in silico database, in silico model, and basically kind of yielded even greater improvements in the quality of the inhibitors being generated. So in other words, we can already have a closed loop system that goes from AI-powered drug design in this case, via synthesis to automated testing and then back into the database. And as I said before, what that means in general is that timelines can be compressed and that research can happen in a fraction of time. And as we all know, within drug discovery, time is money. And anything which helps us to bring a new drug to the next stage faster, or as importantly, to have a drug that should not progress, being identified faster and being sorted out faster, all of that has a massive impact on the likelihood of actually bringing something to market. Because the sooner we can realize that something is not worth taking forward, the sooner we can stop investing into this particular asset, this particular new chemical matter, which is a huge saving for anyone concerned, and then means there are more shots at a potential new drug that can be made. So in other words, what I hope I've shown kind of during this talk is how we can really see the drug discovery ecosystem is evolving from the stage where it's currently at, with its traditional players and traditional approach, to something which embraces AI and embraces robotics and automation, and that when combining those two, we arrive at a point that is even more interesting. So with that, thank you very much for your attention. I'm looking forward to receiving your questions.